Hello, hi all. It's actually a pleasure to be here, and it's really intimidating after all those 2 days. So as Tom already said, I'm really new to the field, so please bear that in mind. So today I'll try to tell you a story about glycobiology of epileptic and healthy human brain tissue and synaptosomes. So over the last 2 days, we heard quite a lot about the fact that glycans are actually implemented in basically all the diseases in the context of any of the diseases, and actually brain is no exception. Not that it's not the exception, but actually there are plenty of the diseases where glycans are implicated, starting from neurodevelopment to neurodegenerative ones, as mentioned here, autism, ADHD, Parkinson's, MS, Alzheimer's, and all the others. And it's not only the pathology of the disease itself, but also glycans affect the neuronal activity a lot. So the main function of the neurons, as we all, I assume, know, is actually to transmit the signal from one neuron to the other. And as you can see here in the slide, the components are resting membrane potential, action potential, packaging neurotransmitters into the vesicles, releasing them, catching them, and transmitting the signal. In all of the stages, you can see that N-glycans are actually marked there, that they are very important and they have extreme function in regulating all the processes, starting from the action potential to the packaging, then releasing, catching, and transmitting the signal. So it's really important to understand what N-glycans do in the brain and what do they do for the neurons. Talking about context of the diseases and the activity of neuronal network, one of the diseases that comes into the picture immediately, it's actually epilepsy. Epilepsy is characterized based on the occurrence of epileptic seizures due to overexcited neuronal network. And there are literature reporting that actually N-glycans are attached to the voltage-gated ion channels, and the very important sugar is actually sialic acid. So sialic acid regulates the threshold of the voltage-gated ion channel, meaning that the more sialic acid, the lower the threshold is, and the excitation of the neuronal activity is easier. The other interesting aspect is that when packing the vesicles with neurotransmitters, here we can see multiple of the pumps, and they all are highly N-glycosylated, and actually they are all highly sialylated as well. And this all comes into the nice picture. In 2007, actually, it was a very interesting study, one and relatively old already, but what the group showed by using in vivo animal model of the rats, so meaning the epilepsy seizure-induced animal models, so that sialic acid has a significant role over there. So once the researchers injected sialidases, the occurrence of the seizure dropped, and then the duration actually dropped significantly. The opposite result The opposite result was obtained once the sialidases were blocked. There were a lot of sialic acid, and then the occurrence of the seizures increased, so does the duration. It all comes into one picture, into the hypothesis that actually N-glycans and sialic acid specifically modulates neuronal excitability. Simply as said, the more sialic acid we have, the higher excitability goes of the network. So in the lab, I actually collaborate with neurosurgeons, meaning that I work with human brain tissue. So I started with study with lectin-ARI, which is easily accessible and relatively easy experimental setup to do. So what I did, I investigated the glycan profile into the control tissue, meaning no epilepsy, and in the epileptic tissue of human brain. As you can see here from the graph, actually there is higher sialylation in the epileptic tissue. So does that there are some other differences in GalNAc, in mannose, and actually in general that there are way more N-glycans than O-glycans, which is not a surprise. But there are some limitations. Lectin-ARI is a very good, very easy and accessible method, however, is not that much specific. And the other thing is that what I got to know here is actually the differences between the tissue. But epilepsy is all about synapses actually. So my question is, what happens at the level of the synapse? There is literature to support that synapses are also highly N-glycosylated. So I decided to move forward and investigate synaptosomes. To do so, what I need to do is actually to extract synaptosomes. So the workflow looks like this. You have the tissue, you homogenize it, and then during a couple of centrifugation steps and sucrose gradient, you have a very nice layers of myelin, synaptosomes, and mitochondria. I take synaptosomes out and I work with this little sample. So to define the human brain glycobiology, human brain synaptosome glycobiology, I started with transcriptomics. So by looking into transcriptomics, first of all, I wanted to confirm that my sample is actually synaptosomes, but not all some trash, so to say, and it was successful. Actually, among top of 500 mostly expressed genes found in the sample, those genes nicely follow the categories of either synapse organization or synapse function, meaning that the dataset is actually the real human brain synaptosomes. Moving forward, I looked into most enriched functional pathways. And over here, there are plenty. The most enriched ones are actually related to the vesicle transport, to the regulation of the transport, to cytoskeleton organization, secretory vesicle, synapse itself, neuronal projections, and all the others, proving that there is actually local transcriptomics happening at the level of synaptosomes. Comparing the groups between epilepsy and synaptosomes, since we are very stringent and precise with our transcriptomics analysis, the threshold is very high. So we found 9 highly downregulated and 16 highly upregulated genes specific for the epileptic synaptosomes. Among those, actually, let's start with the downregulated ones. There are interesting genes such as promoting the organization of cytoskeleton and cell migration. The ones that are actually transcription factors regulating cortical neurons through the cell adhesion, and some other factors with the cerebellar maturation, basically suggesting that the cells migrate less and that the neuronal network probably is even less plastic than control group of the samples. Among the upregulated ones, of course, we found the ones that are related to the vesicle transport, to recycling, to promoting synaptic assembly, neuronal growth. So does the glutamine cycle transporter, calcium permeable ion channel. So basically all the genes that are very actively involved into the activity of the synapse. The next step, of course, was looking into the glycome at the gene level. And here we were happy to see that actually fucosylation is upregulated in the epilepsy. The diseases of glycosylation pathways are also upregulated. So does the glycosyl compound catabolic processes, lipoglycosylation, and the others, actually directly linking the glycome, the glycans, to the disease of epilepsy at the gene level of synaptosomes. Another way of looking at it was analysis of N-glycome specifically. So all the pathways of N-glycome processing is very highly expressed in the epilepsy, so does the couple of the others. Last but not least, of course, we looked into into sialic acid associated pathways, since, as I mentioned, sialic acid is very much actively involved into the opening of the voltage-gated ion channels. And here, not a surprise actually, as expected, all the pathways are very highly upregulated in the epileptic synaptosomes, starting with cellular transferase activity to cellulation and in general sialic acid metabolism. And since I work with neurosurgeons, that means that I get fresh human brain tissue. And that does not limit me to only transcriptomics, proteomics, glycomics, all the other very powerful techniques, but also I'm allowed to investigate enzymatic activity. So what I did, I investigated enzymatic activity of sialidases, and here in the graph you see the the information of CL lidases in the patient's brain, in the cortex and hippocampus of the same patient, meaning that the epileptic core was actually in hippocampus, but in order to remove that, neurosurgeons had to remove a small piece of the cortex itself. So cortex here stands as a control and hippocampus as an epileptic core. And it's normalized in between the patients to avoid the variation, and we can see that in hippocampus, in epileptic core, CL lidases do not do the job, meaning that there's higher CL elation in the epilepsy. Moving forward, with all this having in mind, I realized that this is not enough. I need to investigate glycomics. So then, a year ago, I met Tom and we talked, we established a project, and I arrived to Genos in March, I think, and started a very interesting journey throughout the forest of human N-glycome. So the pipeline looked like this. So I brought my samples of human brain tissue and of isolated synaptosomes. Interestingly, the synaptosome samples match the tissue samples. So from the same patient, I had the tissue and I also had synaptosomes. So we went through the very well-established pipeline, we released the N-glycans, we labeled them. Of course, we performed sialic acid derivatization because that's one of the main interests of mine. And then we move over to the UPLC and mass spectroscopy. Looking at the data, over here you can see human brain tissue N-glycome profile in PCA analysis. So over here you can see that actually it is separated. Control samples are separated from the epileptic ones, and if we look into the epileptic group, we can see the separation between the epileptic hippocampus and epileptic cortex. Then, of course, we looked into the profile of human brain tissue N-glycome. So actually, thanks to this nice database that Borna presented today, even new people like me who just started this journey are actually able and allowed to look what's there, to dig into this huge data set. So we were lucky enough to determine the entire profile of human brain tissue, and then we looked more specifically actually into the differences between the control and epilepsy. So over here you can see that among the 58 peaks, actually in 13, the composition of the glycan is completely different in the epileptic tissue compared to the control one. And while the actually derived traits and the more detailed statistical analysis is a work in progress, for now I can see that even at the preliminary analysis at the level of single peaks, we can already note some differences. For example, high monosis seems to be increasing in the epilepsy, and very interesting observation happens from here that 2-3 seems to be decreasing in epilepsy, while 2-6 is actually increasing. So I do believe that while digging throughout the tail of the chromatogram, the 2-3 and 2-6 linkages, there will be some interesting information found soon. The next step, of course, was to look into the synaptosomes, into N-glycome of synaptosomes. The sample size is a little bit smaller because it's tricky. Not every sample is good enough to prepare the synaptosomes, and also the other thing is that synaptosome sample is very, very small, so it's difficult to perform N-glycome analysis on that one. However, with the help of Tom, actually, we succeeded. So the difference here is that they do not cluster that much, but that's okay, because from the transcriptomics analysis, I also see that transcriptomics actually, there are differences, but they do not differ that much in this term. So by looking at the chromatogram, we already noted that N-glycome of synaptosomes is completely different from N-glycome of the brain tissue, because simply the peak number is different and the overview is completely different. So that's one huge difference. And also, while the analysis is still ongoing, we can already see the post-monosis glycans increasing in epilepsy, and once again, the more differences will be found soon. I'm pretty sure of that. So to sum up my work for today, actually I would like to say that epileptic synaptosomes do display a characteristic local transcription profile, that transcriptomics analysis of epileptic synaptosomes revealed enrichment in diseases of glycosylation and glycan processing and sialic acid turnover pathways, that synaptosomes do exhibit distinct N-glycome profile from the whole brain tissue, and that preliminary results already indicate that glycosylation of brain proteins is altered in epilepsy. So I would like to thank you. I know it's Friday and it's really sunny and nice outside, so thank you for being here. Of course, thanks for the patients, Genos, I'm happy to be here. Thank you, Tom, my own lab, and well, you see all the good things involved in the study. So thank you all. Any questions?